All right. So I'm a student with Uwe Oller. So I want to thank Martin for giving me the chance to speak today. Work. All right. Okay. So the project I'm going to talk about today is basically a collaboration with this corner of the slide. So with the Esteban Mazzoni lab at New York University and the Sean Mahoney lab at Penn State. And really the main person who's been working on this for 2 or 3 years was Sylvia, who produced all the data I'm going to talk about. So the system we're using is direct programming. We have pluripotent cells that we start with, and it's an inducible cell line, so we add doxycycline, and we induce the expression of these 3 transcription factors. And after 48 hours, we get a homogeneous population of motor neurons, of spinal motor neurons that are actually functional and homogeneous. The question we wanted to ask is, why is it that these 3 factors are successful in doing this? Because this is a very short time span, and because we have a success rate, almost more than 90% of the cells turn into spinal motor neurons. So why is it that these factors can do it? And so we did a time course experiment with RNA-seq, ChIP-seq for different histone modifications, and of course for these 3 factors.
Mouse, sorry.
It's mouse. So the starting point, just so that everybody knows, we have in pluripotent stem cells we have these bivalent promoters that have both H3K4 trimethyl and H3K27 trimethyl. So you have both active and inactive histone modifications on the promoter. And the idea is that these genes are poised or are ready to either to become activated. And these are typically the developmental genes. If you actually take in stem cells all these genes that have H3K4 trimethyl and plot the ratio of H3K27 acetyl to H3K27 Me3, you get this nice bimodal distribution. And if you plot gene expression for these things, and if you do gene enrichment, you find that these are the developmental genes that are really bivalent, and that these are the other genes that are actually expressed in stem cells. And so what must happen during stem cell differentiation is that these 2 populations will shift, right? So some of these genes will become deactivated, and some of these genes will become activated, and that this will have to be resolved somehow. So we did ChIP-seq for these 3 marks, H3K27 acetyl, H3K27 trimethyl, and H3K4 trimethyl, over the time course of this differentiation to try to understand how this process is working. So we have 3 histone modifications, we have many genes, and we have different time points. So we want to co-cluster these different histone modifications across time. And we don't know how this is going to look like, so we wanted a very general framework where we can just do this clustering and just look at the data, how it looks like. So it's very simple. The thing we came up with, basically you have something like a Markov chain, or actually it could be a tree. You can, in our case, it's a linear progression because our system is linear, but if you can imagine you have something that has branching, like blood development or something like that, you could also have a more complicated tree. And basically there is, so these things you observe, this is your data, and basically you have the class that then determines how your data looks like. So this is a tree augmented naive Bayes model, except that we don't observe this, and we fit this with an EM algorithm. The main trick is that this is not really the data, but the change in the data. So this, if we have a 0 hours, this is going to be the full change from 0 hours to 12 hours. This is the full change from 24 to 12 hours, and this is the full change from 48 to 24 hours. So this, in a way, becomes more like a second order chain, maybe. And this is nice because they are, when you do the full change, they look Gaussian, but the data doesn't look Gaussian. So this is very good at catching trends, and you can basically have this model. This, of course, describes only one histone modification. You could have a multivariate distribution here to add the histone modifications, the other ones, but actually what we did is we added, so for each new histone modification or each new data that you have, you can add another chain, just like this. So we have three of these chains, all linked to this promoter class, and that's how we do it. So if we run this model on the data that we had on the system I just described, we get 11 clusters. I'm showing you the nicest three here. So the first one, you see that at 0 hours, you had high H3K4 trimethyl and high H3K27 trimethyl and low acetylation, which corresponds to these bivalent genes that a lot of people characterize. And you see that during the differentiation, a switch happens, and you get higher acetylation than methylation at the end. And this corresponds to a very clear increase in gene expression. On the other hand, you get the other opposite side of the coin in a way, where you have things that start high in acetylation and low in K27 trimethyl, and the opposite switch happens, and you get a corresponding decrease in gene expression. And if you actually do pathway enrichment or Go enrichment, you find that these are motor neuron differentiation genes, which makes sense because we're differentiating motor neurons, and these are the pluripotency and cell cycle genes. On the other hand, there are genes that start out like this, so they have, again, this bivalent promoter type thing, and basically nothing happens to them over time, and their expression remains low over time. And these are actually the other developmental genes that are not relevant to our motor neuron fate. So those genes do not get resolved. Yeah, maybe they get resolved later if we kept the neurons going later. We don't know. But as far as we went with 48 hours, they do not get resolved. So this is very good. So we have these different dynamics. If you actually look at a more complete picture, I told you there were 11 clusters. We have 4 clusters that show a little bit, that show like upregulation, and we have 3 that show some downregulation. We have other, so this is 4 and 3 is 7, so we have 4 clusters that basically stay constant, either low or repressed or active. So I'm not showing those here. And basically what you see is that we have actually different types of activation. So not everything looks like the cluster I showed you on the previous slide. We have different trajectories of activation. And also not everything looks like this one here, but we have different trajectories also of repression. And this corresponds to the strength or the dynamics of gene activation. So these are scaled. This is the same data that's plotted here. The gene expression data is just scaled for every gene so that it highlights better the dynamics. And you can sort of see that this is the strongest activation, this is a little bit weaker, and then you see the switch with the other 3 clusters and you start getting the repression. Okay, so this is good. We have different, we have the bivalent motor neuron differentiation genes. They are activated by the switch, and the pluripotency genes are repressed by the opposite switch. You have the bivalent genes that are not related to the motor neuron fate, they stay the same. And we can see that during differentiation, although our system is homogeneous, there is not one trajectory for activation, there are multiple trajectories. Okay, so now we know that that this happens, and we know that we induce 3 factors, and that inducing these 3 factors is what causes this. So naturally, we want to ask, how are these 3 factors doing that? And so we did ChIP-seq for these 3 factors. And we did ChIP-seq at 12 hours, 24 hours, and 48 hours. Because, of course, at 0 hours, they are not yet there. Right, so this is the work from Akshay, who was a student at Sean Mahoney's lab. So what he did is he ran differential peak finding to characterize how these factors behave. And so islet and LHX3, which are 2 of the factors we induce, we found that they always pretty much co-bind together to the same sites. While neurogenin 2 here mostly doesn't bind with islet and LHX3. So you see this overlap, but in most cases, there is not much overlap between neurogenin and islet and LHX3. So basically, we have two types of binding. We have islet and LHX3 binding, and we have neurogenin binding. I should also mention that neurogenin, we only did the ChIP-seq for 12 hours because later on the protein starts getting degraded. So neurogenin is only there at 12 hours. Right, so this happens. So we want to ask, are these factors involved in activation, only activation of the motor neuron fate, or are they involved also in repression, and how do they do this? And one of the first questions we can ask is their binding static or dynamic? Like, do they go to sites that they want to go to and just stay there, or do they change their sites over time? So this is again Akshay's work, and what he did is he ran, again, differential peak finding for LHX3 sites for 12 hours versus 48 hours. And what we get, so to characterize things that are high at 12 hours and low at 48 hours, things that are high at 48 hours and low at 12 hours, and things that stay the same. And since we find big, like large numbers for each group, that means that the binding of our factors is both dynamic and static. We get these dynamic sites, and we get these static sites where the factor goes and just stays there. Right. So since we know all this, one of the main questions that we can ask is, okay, we have the chromatin landscape of the pluripotent cells where we started. Do these factors go to sites that were already accessible in these pluripotent cells, or do they go to sites that were not accessible and they open them by themselves? And so one of the things we did is simply we did a attack-seq at 0 hours at these pluripotent cells, and we plotted the attack-seq counts at the neurogenin sites, so sites that are only neurogenin, and at the sites that are only islet and LHX3. And what we can see, surprisingly, is that neurogenin actually likes to bind more open things than islet and LHX3. We can quantify this by peak finding. So if we do peak finding on this attack-seq data set and intersect the transcription factors with the attack-seq peaks, what we can see is that neurogenin at 12 hours, about 38% of it binds sites that were already accessible at 0 hours. The early only, so these sites here, early only LHX3 and islet sites, about 26% of them bind things that were accessible. The constant sites, these ones, about 10%, and the late sites, very, very few, which makes sense because they come in late during the differentiation. So based on this, we decided to split the first two into two further categories, things that were accessible at 0 hours and things that were not accessible at 0 hours. So we have 6 categories. This, accessible at 0 hours and not accessible at 0 hours, early only, accessible and not accessible, constant and late. Right, conclusion in the middle. So we have neurogenin and islet. Yeah, it's primarily independent. Their binding doesn't overlap except for very few sites. Islet binding is both dynamic and constant. Neurogenin and binding and islet binding happens both in accessible and inaccessible regions. And so if you look at these 6 groups and we plot attack-seq and H3K27 acetyl for these groups over time, what we can see is something really nice, is that when these factors bind sites that were already accessible at 0 hours, these sites get deactivated or decommissioned or repressed, because you see a decrease. So the orange is attack-seq at 0 hours, the black is attack-seq at motor neurons at 48 hours. And you see that these sites, which were neurogenin sites that were accessible, they go down a little bit, and also in the acetylation, if you compare the orange to the black curve, they'll go down a little bit. In LHX3 sites also, that happens. So sites that were early only and were accessible at 0 hours, you see this decrease. While when they bind sites that were not accessible, what happens is that neurogenin causes binding in these places causes activation, so an increase in the active signal. The early only LHX3 sites don't do much. Nothing much happens. The constant sites, they have activation. It's clear from here. And the late-only sites also, they cause their sites to become active. I'm saying they cause it, but we don't know, of course, if they cause it. It's just an association that they bind, and then when they bind, this happens. So, yeah. Right. So then how does this fit? How does this fit with promoters? So I showed you the promoter dynamics, and I showed you what happens to these sites. And and so we assign these sites to their nearest promoters within 100 kb distance. And we construct a random model of associations. And we plot here the log-fold change of the actual associations versus this shuffled random matrix. And what you can see is that promoters that are upregulated here, they are enriched. So red is enriched, blue is depleted. So they are enriched for the constant and late sites of LHX3. But they are depleted. But they are depleted from the sites that bind accessible regions, which makes sense because we know that these sites actually go down in their signal, and we know that these sites, when binding happens, they become active in the actual sites. On the other hand, things that get repressed have very strong depletion of our constant and late sites, but have very strong enrichment for sites that were accessible in EB at 0 hours. So this makes sense because we know these sites are associated with decommissioning of the activity at the actual sites, and they also match genes that become repressed. And these sites are associated with activation of their actual sites, and they match genes that get activated. And those other genes that pretty much remain constant have small enrichment for basically everything, or depletion for everything, so they're more or less random. So this makes sense. And so the next question we can ask is what's happening at these sites that are getting depleted? I mean, these are sites that were active in ES, or in the pluripotent cells we start with, and the binding of our factors happened there, and they went away. It makes sense that these sites were the stem cell enhancers, or the stem cell regulatory regions, and so to check for this, we did OCT4 ChIP-seq at these at 0 hours, so at the pluripotent cells, to check whether we have enrichment of OCT4 at these sites or not. And what you can see is that that's true. So if you compare neurogenin binding to sites that were accessible versus sites that were not accessible, see that OCT4 here is higher than here. And the same for LHX3 early only. You see that OCT4, when they bind accessible sites, these sites had OCT4 enrichment, while these sites did not have OCT4 enrichment, at least compared to that. So that means that these sites were bound by OCT4, one of them in pluripotency network transcription factors. And we don't know, of course, if that's because the sites caused the decommissioning of OCT4, or whether OCT4 was already going away and these sites were just binding to open regions that are just open, so they just bound there, and this is more of an accidental thing. So this needs more experiments to determine which scenario is the right one. So there's a lot more to this study, but I'm not going to talk about it today, just to keep it short. I'm going to make an advertisement for another paper that came out of Uva's lab a year ago. So it was a collaboration with the Jim Kadunaga lab at UCSD. And it's because of the discussion yesterday about promoters, enhancers, which are promoters, which are enhancers, and so on. So this paper is very relevant to this discussion. And basically, if you ask what's a promoter, or what are the characteristics of a promoter, in the promoter you have these sequence elements, which we call core promoter elements, like initiator, data box, and so on. These things lie in a nucleosome-free region that can be big or small, can have variable widths. And of course, on flanking these nucleosome-free regions, there's nucleosomes, and these nucleosomes have different combinations of histone modifications that are sitting on them. You also know that most promoters, or not most, a lot of promoters, in human at least, initiate transcription in two directions for some reason, and one direction is the direction where the gene is, and so these transcripts is what you eventually see in all these RNA-seq studies when you do the poly-A selected RNA-seq. And these ones are very quickly degraded, so you don't really see them in the stable RNA population. So why this happens, we can talk about this. However, what I want to talk about first is you have these histone modifications. You know that transcription goes this way. You have histone modifications here, and you know also there's transcription initiation going this way. So it makes sense to think, okay, maybe these histone modifications are related to transcription initiation that's going this way, and these histone modifications are related to transcription initiation that goes this way. And so we did the correlations. So this, so I should mention the paper. This is the paper. It's mainly the work of Scott Lacady. He's a postdoc at Uwe's lab. And when we did the correlations of K27 acetyl on the nucleosomes that are downstream of the main transcription start site, this is what we call forward, versus the 5' growth-seq, which is a measure of the transcription initiation rate, on the forward and the reverse side. And you can see that K27 acetyl correlates with the transcription initiation that's on its side, but doesn't correlate to the transcription initiation that's happening in the other direction, and vice versa. Yeah. So if you go back to to this one here, these histone modifications correlate with this transcription initiation, while these histone modifications correlate with this initiation, but not the opposite. And this makes sense. So next, if you look at are there different types of promoters, what you can see is that basically you have these promoters which initiate transcription in two directions. One direction makes stable transcripts, one direction makes unstable transcripts. These actually, what Scott found is that these actually come from two different core promoters, so two different sequence elements that are sitting on the edges of the nucleosome-free region in reverse orientation to each other. And what Sasha at Jim Kanonaga's lab did is that he actually showed that these core promoters here can transcribe in vitro. So they are like core promoters, they are promoters which transcribe. So these promoters which are bidirectional or divergent, they're actually the result of having two promoters. Each of them is unidirectional. And when you have this scenario, you have a nice, unique chromatin environment where you have this arrangement of histone modifications downstream of the stable gene. But on the other side, you have a different arrangement where you have basically a chromatin state that has Me2, H3K4 Me2, H3K4 trimethyl, and H3K27 acetyl sitting on the plus one, for this it's the plus one, it's the minus one nucleosome, yeah. So it's sitting right there, while this same state here is actually occurring more downstream, and on the plus one nucleosome here you have another state which only has H3K4 trimethyl and H3K27 acetyl. If you have two what we call bidirectional promoters, which is two promoters the promoter is making two genes, two stable genes. It also results from the same mechanism where you have two core promoters, and results from, and you see the same mirror image of the chromatin environment, so it's different from here. And what we characterize is that sometimes there are open regions where you have only one core promoter going in the stable direction, and you don't see the divergent transcription, and these are what we term unidirectional promoters. And for these, we see some histone modifications on the reverse side, but they don't show a specific enrichment for a specific chromatin state. And usually these open regions are on average smaller than those ones and those ones. Yeah. So this paper is, this is this paper, Molecular Cell, January 2015. There's a review that might come out soon. Yeah, and yeah, that's it. Thank you.
What's the motif? The yellow box in the slide?
It's not really a motif, a certain motif that we found. So we had this sequence model that Scott trained on transcription initiation. Transcription start sites, and this sequence, it's a Kamer model, and basically it learns the sequence of the transcription start site. Yeah, exactly, promoter-like, and then you can scan any other sites and say, okay, this matches these promoters or not, and that's so.
What's the difference between the two types of nucleotides?
Which ones? Here?
Yeah. See, those ones are thicker, and those ones on the left, maybe it's not seen.
Oh no, this is just the drawing. It's just the cartoon is not so good, but yeah, that's just the us not being very good artists.
So is this difference only for
Enhancers have different chromatin state environment, so they have higher ME1 than ME3. And they so enhancers also show this divergent transcription. Some enhancers also show unidirectional transcription. There's a paper from Chris Glass that talks about this. And basically, the transcribed enhancers will have H3K27 acetyl, H3K4. So they will look kind of like this, except that instead of H3K4 ME3, you have H3K4 monomethyl. Yeah.
Maybe no more questions?